The idea that cloning could one day become reality was thought to be a scientific impossibility by many experts. But because of years of research, this science fiction has become reality. Today, I'm going to talk about the processes of producing copies of genetic information to cells and organisms, a process also known as cloning. This picture from Jane Ackery of Injury Board National News Desk shows two mice that have been cloned. There are several types of cloning, including DNA cloning, reproductive cloning, and therapeutic cloning. DNA cloning is the cloning of DNA to copy genes. These genes are used for research. DNA desired to be cloned is transferred into a self-replicating genetic element, such as a bacterial plasmid. It is then placed into a host cell, where it generates copies of the same genes. This image is from Omar Quintero. According to the National Institutes of Health, cloning certain plasmids in order to generate multiple copies of a gene is DNA cloning. The next type of cloning is reproductive cloning. In reproductive cloning, whole organisms are copied. Reproductive cloning is very similar to therapeutic cloning. The main difference is the use of the embryos that are produced. In reproductive cloning, the embryos are transferred into a female host and are developed. In an article, reproductive cloning is the cloning of organisms who have the same genetic information as an existing organism. In this diagram, the nucleus of a particular sheep egg is enucleated, meaning its genetic information is removed. It is then fused with the memory cells of a sheep with desired characteristics. The egg develops and is transferred into a host until it develops into a child. The last type of cloning, which is therapeutic cloning, is a cloning of embryos for research. As the Human Services stated, therapeutic cloning is the production of human embryos for use in research. The goal of this process is not to create cloned human beings, but rather to harvest stem cells that can be used to study human development and to treat disease. Similar to before, therapeutic cloning is very similar to reproductive cloning. The main difference is that the embryos are used in research. These embryos allow for study in human development and stem cells, which are cells able to generate into virtually any type of specialized cell in the human body. In this diagram, courtesy of Wikipedia, it can be used for certain illnesses such as stroke, deafness, missing teeth, or even multiple sites of cancers. An important figure in cloning is Ian Wilmut, the leader behind the research team who developed Dolly the sheep. This sheep was the first cloned mammal. It was also a very big step in cloning because of the new process called somatic cell nuclear transfer. As said by Wilmot, this method of nuclear transfer may never be significantly efficient for use in humans. But because of the research gained from Dolly, many other mammals were able to be cloned. However, cloning is extremely controversial, especially human cloning. Many religions oppose cloning. Also, some believe that cloning only specific parts of the body is alright, while others oppose cloning completely. Areas including the US have banned human cloning. As said by Lester Crawford, an American veterinarian and former commissioner of Food and Drugs, we're aware that there are many social and ethical issues related to cloning of animals. Human cloning is the most controversial type of cloning. Because human cloning is highly ineffective, and embryos are used in the process, many scientists and physicians believe that it is unethical to clone humans. And not enough information is known to be sure of human cloning results. Human cloning is illegal in many areas, including the United States of America. Since Dolly the sheep, many other mammals have been cloned. It's possible that in the future, some animals will have been cloned for their positive characteristics to produce better food products. Also, animal cloning is extremely inefficient, and many cloned animals do not develop into healthy individuals. Cloning may have a very bright future ahead. However, many issues including the fact that reproductive cloning is a very expensive process. Also, Almost all attempts of cloning are failures. Cloned animals also tend to have lower chance of survival.